This lecture video is going to go over the accessory structures of digestion, so the liver, the gallbladder, and the pancreas. Anatomy of the liver. The liver is divided into basically 4 lobes, 2 large primary lobes, the large right lobe, and then the much smaller left lobe. The right lobe is also divided into an inferior quadrate lobe and a posterior caudate lobe, which will be defined by some of the internal structures. It's covered by visceral peritoneum. I just talked about the 4 lobes. There are ligaments that are actually going to, these are folds in the peritoneum that are going to connect the liver to the abdominal wall and to the diaphragm. These ligaments are called the falciform ligament, the coronary ligament, 2 lateral ligaments, and the ligamentum teres hepatis. The lobe is made into units called lobules. There's over 100,000 liver lobules. And these lobules then are the functional unit of the liver. Inside these modules, here's a piece of a module, there are hepatocytes, which are the main liver cell. There is sinusoids, which are going to be specialized capillaries that are going to empty into the central vein. I'll talk about what happens there in a second here. And then there's the portal triad, which is going to include a bile duct, a hepatic portal vein, and then also a hepatic artery. So these are the parts of each unit that make up the liver tissue. Blood flow through the liver. This is called the hepatic portal system. And fascinatingly enough, all of the nutrients that get absorbed into our bloodstream through the small intestines, that blood, before it goes into circulation throughout our body, delivering those nutrients, is going to go through a special portal called the hepatic portal, going through the liver. And so this portal system is like a secret circulatory, small, separate circulatory system that exists, where all of the things that we've done in the digestive videos that we've talked about, all of the blood with those nutrients is going to flow through this secret portal before it goes back into normal circulation. Blood is going to come into the liver through the hepatic artery, which is going to be a branch off of the aorta, bringing oxygen-rich blood to it. Blood is also going to come into the liver through the hepatic portal vein. And the hepatic portal vein is going to bring blood that's slightly deoxygenated, because it is a vein, but it's going to have all of the nutrients in it that was absorbed through the digestive organs. As that blood flows through the lobules and the sinusoids, the liver cells, the hepatocytes, are going to basically absorb all of the nutrient molecules out of there, and at the same time secrete a plasma protein. The blood then is going to leave the sinusoids through the central canal and merge out through the hepatic vein, draining into the inferior vena cava, taking the blood back up to the heart. In addition to nutrients, drugs and toxins are also absorbed, and after processing all of the blood-borne nutrients and toxins, the liver will release the nutrients needed by other cells back into the blood. And like I said, that will then drain into the central vein and through the hepatic vein to the inferior vena cava. So it's a fascinating process. The other thing that liver does is it produces bile. So between the hepatocytes, which are going to produce the bile, are grooves that form are little tiny rooms that form the bile caniculi or the bile caniculus. These small ducts are going to accumulate all of that bile that's being made by the hepatocytes. And from here, the bile first flows into the bile ductules and then into bile ducts. Those bile ducts will then unite to form a really big, large right and left hepatic duct, which will then, they will merge and exit the liver as the common hepatic duct. That duct will join with the cystic duct from the gallbladder, and this forms the common bile duct through which bile flows into the small intestine. So again, this whole structure here is a lobule. You can see the triad that exists. Here's a triad, here's a triad. Remember that the portal triad includes the hepatic artery branch, a hepatic portal vein branch, and then a bile duct. You can see the hepatocytes, you can see the sinusoids, and then you can see here the Kupffer cell. The function of those Kupffer cells is going to be to break down red blood cells. Again, just another picture here of lobule, liver lobule, the portal triad, and one more, another close-up. Here's the histology picture. Again, you can kind of see these sinusoids. This would be the bile duct. This would be an artery. All of these different things would be hepatocytes. So some of the metabolic regulation of the liver, so outside of digestion, would be that the hepatocytes are going to extract nutrients and toxins from the blood. They're going to store and synthesize nutrient molecules, specifically fat-soluble molecules A, D, E, and K are going to be stored by those hepatocytes. They monitor and adjust circulating nutrients, so if high blood glucose sugar or high sugar levels will trigger the synthesis of glycogen, and low blood glucose triggers the breakdown of glycogen and the release of glucose. So the liver plays a role in your blood sugar levels or reacting to blood sugar levels. Some of the blood regulation by the liver, so phagocytic Kupffer cells remove old or damaged red blood cells, debris, and pathogens from the blood. It can help stimulate an immune response if necessary. Hepatocytes also make plasma proteins, which are going to determine the osmotic pressure of plasma, so how much water is in the plasma. It functions as a nutrient transporter, and it plays a really key role in blood clotting and complement cascades, so hormone cascades. So those are kind of the outside functions of the liver besides what the role it plays in digestion, which really is to take all of those absorbed nutrients and put it into the blood. Going back to looking at the role and production of bile, so bile contains water and ions and dilute and buffer bilirubin from destroyed red blood cells. And then it also contains cholesterol and bile salt. The purpose of bile is basically to emulsify fat products or break them down so that they can be further absorbed by the body. And remember, they don't get absorbed directly into the bloodstream. They have to go through those lacteal structures in the villi. So again, this chart just shows some of the major functions of the liver, looking at digestive, metabolic functions, and then other functions. So makes and secretes bile, stores glycogen and fat reserves, maintenance of normal blood levels of glucose, amino acids, and fatty acids, synthesis and release of cholesterol that's found, storage of iron reserves, storage of fat-soluble vitamins. Moving on to the pancreas. The pancreas provides digestive enzymes and buffers to neutralize chyme. It lies right behind the stomach and extends from the duodenum towards the spleen. Its major functions, tissues and functions, the pancreatic islets are endocrine cells that are going to secrete insulin and glucagon, and these will be activated depending on what your blood sugar levels are. Diabetes is a disorder of the pancreatic islet cells. The pancreatic acinar cells are exocrine cells, and they produce digestive enzymes and buffers. And then all of them secrete pancreatic juice. Okay, so the pancreas has a couple different functions: regulating blood sugar, producing digestive enzyme, producing pancreatic juices. Some of the histology of the pancreas then, the acinar cells are organized into pouches called the pancreatic acini. The duct network converges into the pancreatic duct, which will empty into the duodenum. So that's where the small intestines is going to get all of these products from. And pancreatic enzymes are in 4 classes: carbohydrates, lipase, nuclease, and protease. So carbohydrates is going to be enzymes that break up carbohydrates. Lipases are going to be enzymes that break up lipids. Nucleases are going to be enzymes that break up nucleic acids. And proteases are going to be enzymes that help break up protein. Some of the enzymes of the pancreas, I just went over these. Carbohydrates, it digests sugar and starches. Lipase, lipids, nuclease, nucleic acid. Proteases break down proteins. So your pancreatic juices play a really important role in breaking down all of the organic compounds in order to get them to their smallest units to be absorbed into the bloodstream and then taken to the liver. Pancreas has an exocrine function, so again, producing enzymes that help digest our food. And then there's an endocrine function. Those chemicals or hormones that help regulate our blood sugar, specifically insulin and glucagon, but also somatostatins and pancreatic polypeptide. But really, when we talk about blood sugar, we're talking about looking at insulin and glucagon. So how do those pancreatic secretions get released? Well, high acidic chyme triggers the release of a hormone called secretin, and that acts to increase sodium bicarbonate to help buffer that acidic chyme. Then CCK is going to be released, and this stimulates the production and the release of the enzymes, the pancreatic amylase, the lipase, and the protease. Moving on to the gallbladder. The gallbladder is a hollow, pear-shaped organ tucked up under the liver. Its job is to store and concentrate bile. CCK, that hormone, is going to trigger the contraction of gallbladder and relaxation of the hepatopancreatic sphincter to allow bile to enter into the small intestines. And the concentration of bile allows for efficient storage. So when you eat fatty foods, there is bile readily available to help emulsify or break down that fatty food into droplets. Again, you can kind of see here, we have pictures of the pancreas. The interior view of the duodenum shows the opening of where things are entering into. Here is showing you your gallbladder, the common hepatic duct, going again into the small intestines. You can see how that tube is coming into here to the small intestines, and over here would be the pancreas coming into the duodenum of the small intestines. So those are the accessory organs that are going to play a role in digestion.